Okay, good afternoon, everyone. I'm Amrita. I'm doing my PhD in animal biology at University of Hyderabad, and every Tuesday I'm taking this live interactive session for animal biology. Every class I'm introducing myself in case new students are joining every session. So yeah, we are in our week 4, and today, last week also we discussed questions on muscles. Today also we will be discussing some more questions on muscles and concepts regarding it. So basically we are discussing the previous year questions of the assignments given for this course. I'm sure you will be getting, you must have got assignment for every week, and I hope that you all are attending the lectures and solving the present year assignments properly, giving the correct answers. Whatever questions we will be discussing in this live interactive session will revise your concept on the lectures. So every Tuesday, I tell you all that please attend the lectures and then come to this session so that you can answer the correct questions correctly. As this will be an interactive session, I'll discuss with you 10 questions and I'll give you 1 minute time to answer in the chat box. I'm requesting everyone, do not unmute your audios and videos, it will just create disturbance, and we can all interact through the chat box. I am seeing the chat box continuously, and as soon as I give the question, think wisely and answer within 1 minute. Then we'll discuss what is the correct answer, whether you have given the correct answer or not, and then the correct explanation to that answer. We'll discuss all of that, right? And yeah, I want to know, is there any new student in today's session who has not attended the previous sessions? Is there anyone? No one? Everyone has been attending all the sessions? Okay, so there is no new student. Okay, one. Okay, whoever is new to the class, welcome you. I welcome you, and yes, we have completed 3 weeks live interactive sessions. If you have missed the sessions, I think you all must have got a YouTube channel link where I have posted the recorded sessions. You can go there and watch the sessions again if you have missed any. But I'll suggest you all that please join the current class and every Tuesday from 6 to 7 p.m. and we can uh, discuss the questions through this live interactive session. Yes, you can go back and watch the recorded class in YouTube. I'm sure you can do that, but interacting live is something different, right? You will remember what we have interacted. Name. I think NPTEL must have provided you with the YouTube channel name. Is it not? Uh, okay, 1 minute. I'll send you the link of previous year's previous assignment session, and okay, from there you can subscribe to the channel, right? So this is the, yeah, I've sent you the link. So this is the link for week 3's live interactive session. Whoever has missed it can go and see the recording. And yes, okay, so from there you can see the YouTube channel and the live interactive sessions of the previous weeks also, week 1, week 2, week 3, even week 0 is there, right? Okay, it's already 8, 6, 17. We'll start, right? So, yeah, I guess all of you can see my screen, my slides, And I think my audio is clear. So we'll start, right? Okay. So here goes the first question. The sarcomere length dash when actin myosin overlap. The sarcomere length increases or decreases or is unchanged or none of the above when actin myosin overlap. And your time starts now. Start answering. So I'll give you 1 minute time and please note down how many answers you're giving correct and how many answers you're giving wrong. Like at the end of the session, I'll ask you what is your score out of 10. Right? We'll be discussing 10 questions here. So yeah. Okay. This or increases. Some are writing decreases, some are writing increases. Whoever is raising hand, please write down in the chat box if you have any query. We have discussed many things about sarcomere in the last session if you have attended, right? So this is a continuation of those topics. So if you have missed week 3, please go and watch the recorded session in YouTube. S. Madhavan, please mute your audio and if you want to say something, write down in the chat box. Okay. A. 50 students are there and I need answers from 50 of you. Please be active in the session. It's okay if you give a wrong answer. You can know the right answer eventually, but try to participate. Yeah? Don't stay silent and just join the session for namesake. It will be good if we all have a good interactive session, right? So, is everyone done? It's okay if you guess it. If you have not attended the lectures, you will know the correct answer and an explanation also. So that is not a worry. But if you have not attended the lectures, please go back and attend the lecture. That will only help you because that is the motive of joining this course, right? Okay, many people are saying A, B, C. Okay, there are multiple answers, right? Okay. We'll see then what is the correct answer. So the correct answer is decreases. The sarcomere length decreases when actin myosin overlap. Now we'll see how. So here you can see the structure of this. Here you can see we have discussed about the sliding filament theory in the previous session. That is the muscle contraction theory. That is how our muscles contract with the help of these thin and thick filaments, which is actin and myosin. So whenever this thin and thick filaments, that is actin and myosin, overlap, muscle contraction occurs. So what is the sarcomere? From this one Z line to another Z line, the length between the one to another Z line is the sarcomere. And within the sarcomere, there are these thin and thick filaments that are actin and myosin that overlap with each other. Here you can see this is a resting muscle. No contraction is going on. But in B, figure B, muscle contraction has occurred and as you can see the sarcomere length, the length between two Z lines has decreased, right? You can see that after the overlap of the thick and the thin filaments, the sarcomere length has decreased. So the correct answer is decreases. So whoever is given the correct answer, congrats. We'll move on to the next question. Sarcomere length can be related to dash in the myotubes or muscle. Option A, number of nuclei, color of nuclei, tension generated or sodium channels. I'll repeat the question again. Sarcomere length can be related to dash in the myotubes or muscle. Number of nuclei, color of nuclei, tension generated or sodium channels. Think wisely and give the correct answer. Try to give the correct answer. Can you please explain in detail? First question. B. People are saying C. Is anyone having doubt with the first question still? Should I explain it again? Like, I can't start from the basics again, like what is sarcomere, what is a muscle, what is myosin, what is actin. That is not the motto of this session, right? You should attend the lectures and then come to this session so that I can revise you the concepts. I can't start from the basic, like what is a sarcomere, what is actin, what is myosin. I've already discussed in week 3. If all of you have attended the session, you know, right? So we are discussing that muscle system is going on from week 3. Now this is week 4. So if you don't know the basics, please go and attend the lectures. Then you will know it. Like, these questions will help you practice and revise the concepts. And as you answer the questions correctly or incorrectly, and then I'll explain the answer, you will know that yeah, like where you are, like how much concepts you have cleared from attending the lectures, right? So if you're giving wrong answers, you should definitely go back and attend the lectures again and revise your concepts, because we have only 1 hour time and I can't start from the basics, right? So second answer is, most of the people have given answer is C. Okay, let's see if C is the correct answer. Yes, C is the correct answer. So, sarcomere length can be related to tension generated in the myotubes or muscle. It is not number of nuclei or color of nuclei or sodium channels. It is the tension generated in the myotubes. So, let's see how sarcomere length is related to tension generated in the myotubes. Here you can see a graph. Y-axis shows the tension generated in the muscles and x-axis shows the length of the sarcomere. Right? The length of the sarcomere changes as it contracts and relaxes. And based on that, you can see here different stages of contraction and relaxation of sarcomere. Here is it totally contracted. The muscle is fully contracted, and with B, C, D, and E, it is getting relaxed. So, if the muscle is fully contracted, the length is less, and as it gets relaxed, the length of the sarcomere increases. And with this length, the tension also varies, as you can see from the graph here. Like, this is the normal range of the sarcomere in a resting stage. In this, the tension is at this level, and as you see, as the muscle contracts, the tension decreases, and as also the tension, and also when the muscle relaxes more than normal, the tension decreases. So basically, the length of the sarcomere is related to the tension generated in the myotubes. Okay, so we'll move on to the next question. Slow and fast twitch muscle fibers are mostly characterized by. So these are two types of muscle fibers that are slow and fast twitch muscle fibers are mostly characterized by option A, the constituting myosin chain types, the constituting actin chain types, size of DNA, or thickness of the plasma membrane. And your time starts now. Gayatri, please mute your audio. Guys, please mute your audios. Slow and fast twitch muscle fibers. This is a new term. These are two new terms which you must have heard in the week 4 lectures. We have not discussed in the week 3. We'll discuss it today. What are these? What is the main difference between slow and fast twitch muscle fibers? We'll know that. And that is the question actually. Like, what is the difference? How they are characterized? If you see the question, it is not difficult. It is very easy. Just you need a common sense to answer this question actually, even if you have not attended the lecture. Keep noting down your marks, like how many correct answers and how many wrong answers you're giving. And I want answers from all the 50 or 52 students here. Please participate actively. Doesn't matter if you give a wrong answer, it's okay. But try to answer. Try. I'm hoping that you all are able to understand the explanations I'm giving to the answers. If not, you can type in the chat box if you didn't understand some concept. I'll clarify it again. Like I'll try to explain in the best possible way again. If you didn't understand whatever I explained, okay, you can always do that. And if we don't have time here, because this is a very time constrained session, you can always post your queries in the discussion forum of NPTEL, and I will check those discussion forum, and if there are any queries related to this interactive session, I'll definitely try to clarify it. Okay. So, okay. So I think most of you have given the answers. Let's see. It's A or D. Most of them have given A. So the correct answer is A. They're constituting myosin chain types. So the basic logic here was muscle fibers are mainly related to myosin chain types. So these two slow and fast twitch fibers are differentiated because of their different types of myosin chains, right? So we'll see how. So here you can see two types of muscles. There is a drawing here. One is the slow twitch and one other is the fast twitch. So two different types of respiration processes are followed by these two types of twitch muscles. That is, in the slow twitch, oxidative phosphorylation occurs, and in the fast twitch, glycolysis occurs. If we discuss more in detail, then types of fibers are of three types, that is type 1, type 2 and type 2, 2A and 2B. So type 1 is the slow twitch muscle fibers and type 2A and 2B constitute the fast twitch muscle fibers that involve two different types of respiration. So from the name you can guess it. Why is it slow and why the another one is fast? Slow twitch muscle fibers contract slowly. Fast twitch muscle fibers contract at a faster rate. And the slow twitch muscle fiber uses aerobic energy. That aerobic energy means involves ATP, right? That is the oxidative phosphorylation. And the fast contract muscle fibers use the anaerobic energy, that is glycolysis, where the end product is lactic acid. Glycolysis can be aerobic as well as anaerobic, but as the end product is lactic acid, it is a glycolysis related. Uh, they use this lactic acid product glycolysis pathway for their energy. So slow twitch muscle fibers lead to slow fatigue. Fatigue means what? Tiredness. You get tired when you exercise, but if you are exercising slowly, you will feel tired less. But when you are doing heavy exercises, like uh, power sports or something like that, which needs much strength, you will get tired that easily, like, right? You will have fast fatigue. So slow twitch muscle fibers produce less lactic acid. Obviously you are exercising at a slower rate, you are using energy at a slower rate, so the lactic acid produced will be slower. And when you are doing some very powerful sports exercises or sports training, then obviously your energy will be consumed at a faster rate and it will produce more lactic acid. So slow twitch muscle fibers get activated during endurance sports. Endurance sports means which needs a longer time, like running a marathon. Like, right, run in a marathon, what we do, we run slowly, 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 because speed is not related to marathon. We just need to complete our way, right? Marathon is all about that. And in a fast twitch, you can give an example of a sprinters who like give who jump at a higher level and do power sports. They, in that case, the fast twitch muscle fibers in our body get activated. Okay, so we'll move on to the next question. Okay, so the fourth question is, an athlete running a 100-meter sprint would be utilizing the activity majorly from. Okay, I've already given the answer to this. Let's see if you have listened it properly or not. So, options are slow twitch muscle, fast twitch muscle, smooth muscle, or none of the above. Start answering. Yeah. Okay, most of you already know the right answer, and I have already explained that sprint is an example of fast twitch muscle, right? So, if you can see here, a marathoner uses energy from the slow twitch muscles, right? And a sprinter, he uses energy from the fast twitch muscles. So slow twitch muscles use oxygen as a fuel, as they need continuous energy. Like if you're running on a marathon, you will be running for a long time but slowly. So you need continuous energy throughout the process. Okay, so it uses slow twitch muscles. And for fast twitch muscles, you your body provides energy at a faster rate, because you are using the energy at a more faster rate than a marathoner. Do sprinters are example of using a fast twitch muscles. Okay, and you get tired more easily. In that, fatigue comes more easily. Okay, we'll move on to the next question. The force versus time curve for fast twitch muscle is similar to energy output curve of dash, and slow twitch muscle is similar to that of dash. Okay, the question is little big. Please read it carefully and then answer. The force versus time curve for fast twitch muscle is similar to energy output curve of dash, and slow twitch muscle is similar to that of dash. Battery, battery supercapacitor, supercapacitor, or supercapacitor battery. So this is a comparison of fast and slow twitch muscles with supercapacitors and batteries. So many are confused in D and B. Is it battery and supercapacitor or supercapacitor and battery? Read the question properly, then you can definitely guess the answer even if you have not attended the lecture. It is not that difficult as it seems. Late joiners, please try to join in time. People are still joining and already half of the session is over. So please try to be on time. Okay, I'll go on to see what is the right answer. I hope most of you have given the answers. So the correct answer is supercapacitor and then battery. So the force versus time curve for fast twitch muscle is similar to energy output curve of a supercapacitor, and slow twitch muscle is similar to that of a battery. Like this we can easily guess. I have already explained what does fast twitch muscle do and what does slow twitch muscle do. The supercapacitor gives energy at a faster rate, if you all know the basic science, right? And batteries go on for a long time and they act at a normal range. That is the activity of a slow twitch muscle. If I want to show you by a picture, here you can see. So this is for the, this is the force versus time graph as given in the question for fast and slow twitch muscles. So you can see for a fast twitch muscle uses force at, like suddenly it uses and then goes down. It is not continuous for a long time. The time period is very less, right? Similar, but the slow twitch muscles use the same force but at a longer period of time in a continuous manner. I guess till now you must have understood the basic difference between slow and fast twitch muscles. If we compare these two muscle types to a capacitor and battery, obviously a capacitor works at a more faster rate, it gives more voltage than a battery. As you can see, it uses more volts from the start and then it goes down, it just declines. But battery, it starts with a faster rate but then it doesn't go much less, it remains a saturated level for a very long period of time and then goes down, just like a slow twitch muscle. So I guess you can understand this comparison well through this, both the graphs. Okay, so we'll move on to the next question. Dash acts as a stable and readily available source to generate ATP and hence energy in muscle. So the options are creatine phosphate, creatinine phosphate, calcium or sodium. Which of these four acts as a stable energy for muscle? Start giving the answers. Think wisely. Do not be confused between creatine phosphate or creatinine phosphate. Okay, many of you are saying A, some are saying B also. So everyone is sure it's not calcium or sodium, right? Okay. I'm hoping you all are giving correct answers after attending the lectures. If any of you are feeling that my explanations are not up to the mark or you are not understanding it well, you can definitely write in the chat box. And if you are able to understand it well, it's well and good. But like, do not go out from this session with a confused mind. I want all of you to be clear with all these 10 questions and the concepts behind them. The present year questions will also have similar questions to these questions, and after attending this session I think you will be able to answer those questions correctly. You will be able to think wisely after thinking the answers for these questions, right? Okay, most of you have given the answers, I guess. So the correct answer is A. So whoever has given the right answer A, that's really great. So creatine phosphate acts as a stable and readily available source to generate ATP and hence energy in muscles. So creatine phosphate is the stable source. We'll see how. So there is a resting muscle which has ATP in it, but it is so there are energy contractions, energy for contractions, like obviously all muscle contractions need energy, and for that they need ATP molecules present in the muscle, right? So muscles tend to have limited quantities of ATP, so they need ATP molecules. So the ATP molecules get used up instead of resynthesizing more ATP molecules. Instead of creating more ATP molecules, first of all, what happens? The ATP molecules present in the cell, the limited quantity gets used up by the muscles. So that's why the creatine phosphate present within the muscles is also a high energy compound that breaks down and makes more ATP. So you can see there is ATP, and when creatine binds to it, it becomes creatine phosphate and releases ATP. So the with help of creatine kinases are that act on phosphates. So with the help of these creatine kinase, the active muscles get ATP from this creatine, right? In the resting muscle, what happens? The creatine combines with this ATP to form creatine phosphate and ADP. And during contraction, when the muscle starts contracting, the creatine phosphate breaks into creatine with the help of this creatine kinase enzyme. Kinases do what? They act on phosphates, right? This is a basic action of a kinase enzyme. So what happens? Creatine kinase acts on this creatine phosphate and breaks it down into creatine and ATP, which is the main energy source for muscle contraction. So you get more energy than the limited quantities of ATP. So creatine phosphate is the one which helps in, which is a stable source for ATP in the muscle. I hope everyone understood it. We'll move on to the next question. Which of these is not a component of the spinal cord? Which of these is not a component of the spinal cord? Hippocampal neurons, sensory neurons, motor neurons, or interneurons? Your time starts now. Start answering. If we if we take creatine in a preferred amount, does it get deposited and interfere in our filtration process? Preferred amount in the sense, yes, if you take more than needed, it may interfere with your filtration process. If you consume it from outside, is it what that you're saying? Like the body creates that much, whatever creatine is present in the body, uh, that is utilized well by the muscles for creating ATP. If the muscles have insufficient ATP, then creatine comes there in that process and provides the extra ATP whatever is needed by the muscles. But if you consume creatine from outside, like if you have a creatine deficiency or something like that, then it may not be, it may be the case then that it won't harm you. But if you take creatine from outside and it is overdosing in your body, then yes, it may affect your filtration process as well. Okay, has every one of you given the answer to seventh question? Which of these is not a component of spinal cord? A. Is everyone saying A? Okay, we'll see what is the right answer. Yes, hippocampal neurons. So, hippocampal neurons are the ones which are not present in the spinal cord. As the name says, hippocampal neurons are present in the hippocampus, which is a region of brain, and it doesn't come under spinal cord, right? So spinal cord has these three neurons, that is the sensory neuron, motor neurons, and interneurons, that is also called as these relay neurons, etc. Okay, this was a very simple question. We'll move on to the next question. In the spinal cord, ascending pathway is ventral pathway, sensory pathway, motor pathway, or none of the above. In the spinal cord, ascending pathway is ventral pathway, sensory pathway, motor pathway, or none of the above. Start answering. I am not sure if I'm getting answers from all of the students. I'm thinking some of you are not answering. I'm requesting again. Please try to answer. It's okay if it's a wrong answer, because I'll be telling you the right answer, but not participating is not a good idea. Okay, most of you are saying B, sensory pathway. Is it? Is everyone done answering? Should I say what is the right answer? Okay, okay. So the correct answer is B. Good. Good. Whoever has given the correct answer. In the spinal cord, there are two types of pathways, that is the ascending pathway and the descending pathway. As you can see from the diagram here, ascending pathway is also known as the sensory pathway, and descending pathway is the pathway which is also known as the motor pathway, right? So pathways are either ascending or descending. So what is the main difference? That ascending pathway carries information from receptors to the brains, right? So ascending pathway carries information from the receptors to the brain, and the opposite happens in the descending pathway, that is they are conveying information from the brain to the spinal cord. Okay, so this is the motor pathway, and ascending pathway is the sensory pathway, as it contains sensory receptors which carry the information to the brain, whereas there are no kind of receptors in the descending pathway. The brain conveys the information to the spinal cord through the motor pathway, okay, or the descending pathway. So we'll move on to the next question. If load is applied on a skeletal muscle which equals to maximum force that can be exerted by the muscle, velocity of contraction becomes dash even when muscle fiber remains dash. Read the question carefully and try to answer. If load applied on skeletal muscle equals maximum force that can be exerted by the muscle, velocity of contraction become dash even when muscle fibers remains dash. Options are high activated, low deactivated, infinite deactivated, or zero activated. This may seem a little confusing. Read the question properly and then try to answer. Everyone, please do not stop. If you are thinking it is a difficult question, try to answer, try to think and answer. Please participate. That will only help you. I'll discuss the questions and I'll go, but it's up to you whatever you are gaining from this session, right? This is all for your sake. So try to be active and answer. It's okay if you're incorrect. Everyone is answering A or B. Okay, let's see. Okay, someone said D, Archie, or else everyone is thinking A or B. Maximum if you are thinking A, high activated. Okay, let's see. So the correct answer is D. I guess only one of you has given the correct answer. Very good, Archie. Okay, so the answer is if load applied on skeletal muscle equals maximum force that can be exerted by the muscle, velocity of contraction becomes zero, even when muscle fiber remains activated. So what happens if the load in the skeletal muscle, if you put a load on the muscle and it becomes equal to the maximum force that can be applied on the muscle, then the velocity of contraction becomes zero, that the muscles will not contract even when they are activated. It is like your body, we have a body, right, and we have a maximum capacity of working. If you put extra load on your body for working, like if you've given extra load to your body, it can't take all the load. It will take till it reaches a saturation point, like the maximum force, and then it will be zero. Even when your body is activated, you won't be able to work because you have reached that saturation stage. If I want to say with this diagram, you can see here, this is a graph of the velocity of contraction with respect to load opposing the contraction. So as it is written here, skeletal muscles contract extremely rapidly when it contracts against no load. If there is no load on the muscles, they contract extremely at a very high speed, and the velocity of contraction becomes progressively less as the load increases. As our body gets more load, the velocity of contraction starts decreasing. The velocity of contraction starts decreasing as the load increases. Okay, these two components, velocity and load, are inversely proportional to each other. Inversely proportional means if one component is increasing, the other will decrease, right? If load increases, velocity of contraction will decrease. So when the load has increased equal to the maximum force, right, equal to the maximum force, then the velocity will become zero. The velocity of contraction will become zero and no contraction occurs even when your muscle fiber is activated. If you have put load to your muscle which is equal to the maximum force that can be exerted to that muscle, it will stop contraction and although the muscle fiber is activated. Okay, so we'll move on to the last question. Okay. Okay, sorry, this is the 10th question. I've written as 9. The active tension dash as the muscle is stretched beyond normal length, which is when sarcomere length is dash. I've already shown. Okay. Try to think and answer. The active tension dash as the muscle is stretched beyond normal length, which is when sarcomere length is dash. The active tension decreases or increases as the muscle is stretched beyond the normal length. If you stretch the muscle beyond a normal length, what will happen to the tension? Will it decrease or increase? And what will happen? What is the sarcomere length at that time? 2.2 micrometer or 1.65 micrometer? Okay. This is the last question. This is question number 10. I have written it as question number 9 by mistake. So, okay, most of you are giving the answer B. I already clicked on the answer. Okay, it's okay as long as you understand the answer. So, the correct answer is B. The active tension decreases as the muscle is stretched beyond normal length, which is when the sarcomere length is 2.2 micrometer. We'll see how. So here you can see. So the first of all, the basic information is the resting sarcomere is about 2.2, sorry, it is micrometer, 2.2 micrometer long. The resting sarcomere has a length of 2.2 micrometer long. It is not millimeter, typing mistake. If the sarcomere is overstretched, if you stretch the sarcomere more, then what will happen? The number of cross bridges that can form is reduced linearly with increasing length. Like the thick and thin filaments which are overlapping each other in the sarcomere, if you stretch the sarcomere, the overlapping will decrease, right? The overlapping will decrease and the length of the sarcomere will increase. Then what will happen? It is obvious that you can see from the cartoon here, you can see from the graph here that the actin filaments do not overlap the myosin filament if you stretch the sarcomere. So no cross bridges form. So there is no contraction. What will happen if you stretch the sarcomere long? There won't be any overlapping of the thick and thin filaments. And that is the basic concept behind muscle contraction, that the thin and thick filaments should overlap, right? So if there is no overlap, there is no muscle contraction, and as a result, the active tension decreases. If there is no muscle contraction, if there is no contraction, the tension also decreases, that is the force decreases. If the length becomes more than 2.2 micrometer, as you can see, this is the 2 micrometer length and here is the 2.2 micrometer length, and if the length goes beyond this point, the force decreases. You can see here the force decreases, that is the active tension in the muscle decreases because it is not contracting at all, right? So with this we finish our 10 questions here. Now I want to know your score, like how much score you got out of 10. Please be honest. I just want to know how much score you have got. 8. Okay. Today's question were a little difficult if you have not attended the lectures, like it was more on a mathematical side, or I will say, or yes, it was more deeper in the muscle physiology part. So only 4 to 5 students are answering. The rest of you have not answered, or rest of you didn't count the scores. What is 7 by 1? 7 by 10, I guess. Okay, so the highest score is 8. No one has got 9 or 10. 10 out of 10, no. As well as 9 also no one has got. So the highest score is 8. Okay, someone has got 9. Great. Even 8 is not a bad score at all. 8 is not a bad score at all. It is very good. I am happy that you all are answering actively and you have got such good marks. And keep up the spirit. And as I always say, keep attending the lectures, then come to the class. All the questions discussed here are all discussed in the lectures. So if you have attended the lectures and understood them in detail, you can get a 10 on 10. No one can stop you, right? Okay, so it's already 7. We'll stop here. We'll meet again next Tuesday. And if you have any queries, please type in the discussion forum. And if you want to see the lecture, this interactive session again, you can go to the YouTube channel and see it. Okay, so we'll meet again next Tuesday. Till then, keep learning. Thank you all. Thank you. Okay, bye.